Hey, hi everyone. We will start the talk session now. First of all, thank you for taking time to tune in our talk shop session today. And my name is Jia Xuan. I'm from the Outreach and Education Unit of the Lee Kong Chian Natural History Museum. Today, we are very happy to have with us Mr. Clement Ng, a PhD student at the Asian School of the Environment, Nanyang Technological University. Tonight, he will speak to us about his research work on fish otoliths or fish ear bones or ear stones. And he will also elaborate on how fish otoliths can inform scientists on a wealth of information that can help them to further understand the ecology of Singapore's fishes and develop tools that help us better manage our food supply as well. So before we start, here are just some gentle reminders to take note of for today's session. So please take note that today's session will be recorded and uploaded to the Education Resource tab of our museum website approximately within 2 weeks from today. And the session duration is expected to be around 45 minutes long, including the Q&A segment. But depending on the number of questions we get, the session may be slightly longer than 45 minutes, but it should not exceed 1 hour. Throughout the session, we would like to request for you to submit any questions via the Q&A icon that you can find at the bottom of your Zoom panel instead of posting your questions for our speaker in the chat box. So if you use the Q&A function in Zoom, you can also view questions submitted by other participants and upvote questions that you find interesting and would like to know the answer to. So Clement will be speaking for about 30 to 40 minutes, and any questions submitted will only be answered after that during the Q&A segment. If you would like to ask your questions verbally during the Q&A segment instead, you may use the raise hand function that you can find if you click on the reactions button at the bottom of your Zoom panel, and I will assist to call out your name to ask your question. Lastly, we would highly encourage you to stay till the end of the session to fill in our feedback form. A QR code will be flashed after the Q&A segment, and you can actually suggest topics you would like us to cover in future seasons of our museum's Talkshop series. If you would like to enter our giveaway contest to win a set of exclusive Museum Marine Biodiversity of Singapore postcards, please enter your email address in the feedback form. Entering your email address is optional for filling up the feedback, but compulsory for the giveaway because the winners will be contacted via email. We will be giving out 3 sets of postcards for today's session. With that, I will stop sharing my slides and will let Clement take over, and I hope everyone enjoys tonight's session. Thank you.
Hi, good evening, everyone. Welcome to today's talk shop session. I'll just quickly share my screen. Great. I think hopefully everyone can see my slides now. So hello, everyone. My name is Clement, and welcome to today's session of the talk shop. Just a quick self-introduction. I'm Clement, and I'm a PhD student at the Asian School of Environment based at Nanyang Technological University of Singapore. So I am trained as a fish biologist, so that's my training, and my research focuses on the status and future of marine capture fisheries from the Indo-Pacific region. So imagine you're a fish living in the turbid mangrove waters of Singapore. You can barely see in these murky waters, and potential predators are lurking in every corner. But you're hungry as well, and you need to eat. You may be thinking of your sense of vision, but the waters are murky, which makes it difficult to see. Perhaps your sense of smell, but the waters may be polluted, so it will occlude your sense of smell in these waters. How do you navigate these environments? One sense you may not think about is the auditory system, or the sense of hearing. So now I know what you might be thinking. Wait, fish have ears? How do they look like? Do they look something like this? Massive ears? Or do they look something like this? Humans have this flappy external ear structure known as the pinna, which helps to capture the sound waves, but also an inner ear system which converts the sound waves into electrical signals sent to the nerves and to the brain for us to perceive as sound. Likewise, fish also have inner ears, and they rely on their sense of hearing to navigate around the ocean or marine or freshwater environments. What I am interested in is less regarding hearing, which is not my main research focus, but a tiny mysterious stone found in the ears of fish. So they look something like this, and they are known as otoliths. So otoliths are tiny bone-like structures found in the inner ears of all bony fishes. These structures are made of calcium carbonate, so the same material you get as seashells, as well as eggshells. Here we have an image of a fish with its otoliths located within its skull. The first image on panel A shows a whole fish, and panel B shows an X-ray image of the fish. The third panel shows where the otoliths are, and each fish has got 3 pairs of ear stones. So these ear stones play a crucial role in fish hearing and balance, as well as orientation in the aquatic environment. Just as a fun fact, the word otolith is made up of 2 parts. Oto is the Greek root word for ear, while lith is the Greek root word for stone. So a question you might ask is, why would anyone be interested to study a stone found in the ear of the head of a slimy fish? So that's because these structures have very useful properties. So all fish deposit material into their otoliths daily, which grow continuously throughout the life of the fish without being reabsorbed. This provides us as fish biologists a continuous and permanent growth record of the life of each individual fish. These unique biological properties then provide us with valuable information on the life history patterns of each fish. Collectively, we can not only study the individual, but the population, which is the sum of all individuals, the community, which comprises of all living organisms, as well as the ecosystem. So this comprises of both living and non-living organisms, which form a marine or freshwater system. So one useful application of otoliths is studying the age and growth of fish. So fish are like trees. The figure on the right shows an image of a tree trunk with banding patterns indicating the age of the tree. Similarly, otoliths produce alternating opaque and translucent annual growth bands, which look something like this. So the otolith grows outwards from the core, which is the timing of birth, and it grows outwards towards the edge, which is the time of death. This allows us to determine the age and growth rates of fish, similar to tree rings. So here's an example of a fish I worked on previously. We have the Australian snapper, a very delicious fish that was caught in Victoria, Australia in November 2014. Basically, we extract the otolith and cut a section through the middle, which contains the core and represents the point at which birth begins. And we look at the cross-section under a microscope. Here, we know that the fish was caught in 2014, and this is indicated on the outer margin of the section. Next, we count the number of growth bands along the section, as well as the core, and this allows us to estimate the age of this fish to be about something like 10 years old. So as the otolith grows outwards along all axes, we can verify this reading by counting the number of growth bands along a different axis. So here we can also see that there are also around 10 growth bands. So we can safely say that this individual is 10 years old. So let's try guessing the age from a different fish. This fish is known as the orange roughy. It's about 1.5 kilograms, so about less than half of the previous snapper that we had before. It lives in depths of about 500 to 1,000 meters, and it eats shrimp, fish, and squid. Here's how the otolith section looks like. Does anyone want to fathom a guess on how old this fish is? Feel free to just shout it out, or if not, I'll just call Jiaxuan to answer for me.
Someone has said 200 years.
200 years, that's a good guess.
And Eugenia says 100 years.
100 years, cool. So scientists have found that the orange roughly lives up to about 250 years old, and they mature at about 30 years old. This makes the species really vulnerable to overfishing. So let's quickly take a step back to understand why it is important to understand fish age. I have a quick case example on the orange roughy. So previous research in the 1970s by some fishery scientists estimated the fish age to be about a maximum of 30 years old. But later in the 2000s, scientists realized that they grow up to 250 years of age. That's way more than the previous estimate. And they mature at 30 years old. So this lack of understanding of fish biology has resulted in unsustainable fishing practices during the 70s. And this has resulted in the collapse of the fishery in New Zealand, but subsequent closure of the fishery has led to its recovery. So let's go back to the alternating annual opaque and translucent growth rings. So these growth bands are based on seasonal changes in the environment. So in temperate countries, this would be related to summer and winter temperature patterns or seasonal climatic patterns. However, in Singapore and the broader Southeast Asian region, these growth patterns are likely related to the wet and dry seasons or related to our monsoon patterns. So the fact that these year stones grow continuously as the fish ages and do not get reabsorbed into the fish provides us with this useful, valuable life history information on each fish, which we can study and extrapolate to populations. Let's make another guess. So this is a species that I'm working on. This species is known as the Malabar blood snapper, and it's actually one of my samples that I collected for my PhD. So it was caught in 2020 and from the Riau Archipelago, where Singapore imports most of our fish samples. So it's sold in our local fish markets, commonly used in our curry fish heads, and it's also about 4 kilograms in weight. So here's a cross-section of the otolith. And does anyone want to fathom a guess on how old this individual is? So it's a real-life sample collected in the markets of Singapore, actually.
So we have one participant that says 15 years old.
15 years old.
And
a bit on the high side. Yeah.
On the 10. And
Okay.
one answer says 9.
Yep. So we know that this sample is collected in 2020, and you can look along the growth axis. So it requires some practice and some training to know where these growth bands are. So I would say it's 6 years of age. So you can also look at it on a different growth axis. And you can tell that this fish caught in 2020 was born in 2014, and you can see 6 alternating opaque and translucent growth rings. So we as fish biologists, we not only look at the alternating growing patterns along one axis, but we count it along different axes to make sure that they are consistent and that we are confident in the age of this fish. Additionally, we can measure the distance between each growth band, so between each age, and this gives us a proxy for the growth rate of the fish. So having knowledge of the capture date and location, as well as the fish size, allows us to get a glimpse into the age structure as well as the growth rates of fish populations. And this would in turn inform and allow fishery managers to estimate, for example, the maturation age of fish, the mortality rates, as well as the population size of a particular species. Ultimately, what we want to achieve in gaining a better understanding of fish populations is to provide sustainable seafood and healthy oceans for generations to come. So I work closely with fishermen from the region, and coupled with this knowledge of where they are collecting their fish, as well as this knowledge on the age structure of populations, we can actually assess whether a population is currently overfished or whether it's being managed adequately. So if populations are being overexploited, we can consider imposing, for example, quotas on how many fish can be captured per year, or provide restrictions on the gear limits or size limits, or close the fishery to ensure that populations will not crash. This would consequently provide us with the seafood that we enjoy and provide jobs for many coastal communities that rely on fishing for a living. So we can link this understanding of fish populations to environmental impacts as well as factors, like studying the effect of climate change on fish. Humans are drastically altering marine ecosystems, yet we have a poor understanding of how climate change affects fish populations in the tropics. So we often hear about fish under migrations from hot to cool waters, but these examples are often not from the equator. So that begs the question of what is really going on with populations from the region. So my work as a PhD student and my research focuses on understanding the growth drivers of fish populations, so specifically on tropical snappers from Southeast Asia and the broader region. So I'm linking these environmental variables to fish growth patterns. So here's a graph describing fish populations on the y-axis and time on the x-axis. We have growth indicated by the blue line, and we are trying to link a biological pattern, such as growth, to an environmental variable of interest, for example, temperature. So by studying how fish growth patterns change over time, and we can link it to an environmental variable like temperature, we can really identify and assess if our local food supply will be impacted under future climate change scenarios. So what I have found in my study was that tropical snapper growth was mainly affected by ocean temperature, as well as the Indian Ocean dipole, which is an ocean-atmosphere interaction that influences the regional climatic patterns. So this represents the first long-term reconstruction of fish growth patterns from the Southeast Asian region, as well as from the equator globally, actually. So, but a question you have in mind may be that fish move. So how can we safely associate an environmental variable to something like fish, which can swim about and do their own things? And this brings me to my second point, which is on trace elements and isotopes. So as fish grow, they incorporate elements from the surrounding environment and their diet into their otoliths. So these elements substitute for calcium in the otolith structure itself, knowing that this is made of calcium carbonate. And so this natural biological substitution provides a unique chemical signature along the otolith growth axis, which reflects where they were in space and time. So we can see, based on the figure on the right over here, that in the first panel, we can see the annual growth bands that is occurring in this Amazonian fish. And in the second panel, we see measurements of the element concentration in the otolith axis. So we can see stronger colors or bright yellow colors, which indicate the fish is moving into fresh water. So by aligning it up to the age of the fish, we can really tell that, okay, this fish is actually in fresh water, and then it moves into the marine environment, and then it goes back into fresh water, and so on and so forth. So analysis of the elemental profile of this otolith allows us to ask a lot of questions. So we can use it to study the natal origins of the fish, so where the fish was born, as well as the pollution history of fish. So whether a fish is swimming in a polluted water, and you'll get detected in the otolith itself because we know that it's not being reabsorbed. Additionally, if we can link water chemistry to the otolith element profile, we can establish where movement patterns occur. So when you think about fish that move, the first or one of the first species that may come to mind is salmon. We know that salmon undergo their migration as juveniles from freshwater to sea, and they grow up into adults where they mature before they return into the rivers to spawn. And so we can dwell deeper into our case example on salmon in Alaska. So scientists can measure the concentration of elements along the otolith growth axis. And we can link this to known water chemistry levels to sort of describe movement patterns across space and time. So the scientists in this study, based in Alaska, measured the water chemistry and grouped them into various unique chemical signatures. These are indicated by the various colors on this plot over here. So they then measured the chemical levels on the otolith surface. In this case, it's strontium, which is an element, and which allowed them to predict where fish were moving, as shown by this circle. So this is an example of how they would move across space and time. So it would be living in something like the isotope group 7, and then in this year, it decides to undergo its long migration into the marine environment, as you can see from the x-axis. So what are some other applications of otoliths? We can also use otolith shape to study fish. So otoliths come in all sorts of shape and sizes, which vary between species, habitat use, and with age. So fish that live in more complex habitats tend to have larger otoliths. So what I'm holding is a deep water snapper, commonly known as angoli in our local markets, and it's commonly used in our curry fish head dishes. It has relatively large otoliths, so I've actually got one here. So this is the otolith of a snapper, and it's about 2 centimeters for this individual. In comparison, the kuning fish found on the plate of nasi lemak has smaller otoliths. So you have a kuning fish that looks like maybe 5 centimeters, deep fried and with your coconut rice, and the otoliths would be about 4 millimeters in length. So 1/5 of the size of the previous species. But fish size is not necessarily a good proxy for otolith size. The mahi-mahi I'm holding is about 1 meter long, so 7 times the length of the kuning in the previous photo, yet its otolith is half of its size. And it gets really tricky to extract the otoliths when it comes to smaller species, for example, pufferfish. So this variation in otolith size is related to their families. So certain families have larger otoliths compared to other families. So the pufferfish here is about 6 centimeters. And these otoliths, which were dissected a few months ago, were about 0.5 millimeters in length, which makes it really challenging to dissect and extract. So what's the application of understanding about otolith shape? We can use fossil records to discover new species. Scientists have also extrapolated fossil records of otoliths to understand the relationship of how big fish were during prehistoric times. Or you can use otoliths to distinguish species. For example, you can use otoliths to differentiate similar looking species across life stages that are in the same environment. One example is a study that compared the juveniles of 2 morphologically identical species using otolith shape. The scientists or the researchers found that they could actually differentiate the species based on how different the otoliths look under a microscope. Finally, you can also use it to reconstruct the diet composition of ancient human communities. So there was a study that looked at Viking communities from Iceland. The researchers dug up middens which are ancient food waste dumping grounds, and looked at the otoliths in these areas. They were able to match the otolith shapes to species that were being consumed by the Vikings, and based on the sizes of the otoliths, they were able to identify the size of the fish in the waters at that time, as well as understand what ancient human communities were consuming at that time. So it's crossing between fish biology as well as anthropology, as well as history. In Singapore, we have a poor understanding of the ecology of fishes. Situated on the equator, we do not yet know how climate change, for example, warming oceans, ocean acidification, will affect our native fish populations, and in turn affecting, for example, the recreational fisherman community, as well as our food supply. So the work of my lab that I'm in, of the lab that I'm in, aims to fill this knowledge gap to better understand the ecology of fishes so as to safeguard our food supply into the future. So we have a global perspective based in Singapore. So each colored circle on the figure on the right here represents fish that were collected for our study in Singapore. So you can not only look at individual fish populations to get a better understanding of fish life history patterns in Singapore. For example, within each reservoir, how old do each fish get? So broadening our perspective, we can also gain a better understanding of fish movement patterns, for example, by analyzing trace element concentrations in the otolith surface, which will inform us how we can establish conservation areas. So we have ongoing work in our lab assessing when fish migrate between brackish water, so a mixture of fresh and salt water, into a fully marine environment. And zooming out even further, we can understand how climate change affects fishes across the Indo-Pacific region. So one of my current studies is looking at over 1,000 fishes to get a representative sense of the effect of climate change on fish populations. This has implications for our food supply in Singapore, where we import more than 90% of our fish from neighboring countries. It also has implications for other nations, so our research can provide them with a better understanding of how climate change may affect their fish populations. So in summary, otoliths are bone-like structures found in the inner ears of fish. And the analysis of the annual bending patterns allow us to determine fish age as well as growth rates. And trace element concentrations that are measured from the otolith growth surface allow us to estimate movement patterns. And we can use this understanding to get a better and more comprehensive knowledge of populations, the community, and ecosystem level processes. So I'd like to thank you for your attention, and I would be more than happy to take any questions. Thank you.
Thank you so much, Clement. Okay, so I will assist to read out some of the questions from our audience. So from the Q&A section of the Zoom panel, there are two questions. One of it is from May. Is the shape of the otolith unique to the species of fish?
Yes, it is. It's very unique to the species. So each species has got its own unique shape, and you can, so I've got one over here. It's about 2 centimeters in length, and it looks very different to something that is of the same age, but it will be much smaller in size. So we can use it to distinguish between different species and different groups of species, like families.
Mm-hmm. And the next question, do larvae of fish also have otoliths?
Yeah, that's a great question. So the larvae of fish actually have got otoliths, and let me try to find a photo. Okay. So this is an example of a a larvae that still has its yolk sac, and you can actually see the first two otoliths being formed within the head of the fish. So it's I think it's formed, it varies between species, but in general, it's formed within a month of it coming out from its yolk sac. So yes, the answer is yes, and it provides a unique growth signature throughout its life.
From Cleo, what is the advantage of using otoliths to determine age as compared to using fish scale?
Okay, that's a very technical question, and I love it. The difficulty in using fish scale is that scales are being reabsorbed into the system, the body of the fish in times of malnutrition. So you may not get a full or complete proxy of its entire life, unlike an otolith where once it is deposited, it will not be reabsorbed into its body. But great question.
We have a lot more questions. So this is from Jing Yi. Do otoliths have other uses outside of helping the fishes to hear?
Based on my understanding, it's no. So they primarily use their otoliths to hear in the environment. So they have got little sensory cells that help to sense the vibration. And these otoliths help to detect when the fish tilts and when it swims and when it turns between its axes. So that's its primary function within a fish.
Are the otoliths from certain species in the same family often more similar to each other?
Yep, that's also a great question. It's more similar within a species, within a family, sorry. But you can tell based on, you can look under a microscope and you can do a shape analysis to compare differences between each family itself. Yeah. So this is an example of how it might look like between different families. So you've got some otoliths that are more oval in shape, while others are long in shape. So I've got one more example based on that, where so you have got fish of different groups and they look slightly different. Some are more rounded, some are more elongated. And all of these were collected in our local waters, a mixture of fresh and salt water.
Okay. Any differences in otoliths raised in aquaculture versus in the wild besides mineral composition?
Actually, is okay. That's a very good question. I was in an otolith conference about 2 weeks ago, and there was a really interesting talk where they compared the otoliths of aquaculture fish as compared to wild fishes. So they had sort of a roundish tank, and the fish were swimming in the tank. So they noticed that the development of one side of the otolith was always a bit deformed when it swam in one direction. So imagine if you swim clockwise in one direction, the otolith on the inner side was always more deformed as compared to fish that were swimming in both directions. So if you alternate the water flow, so it actually affects how the fish hear in their environments. And it was detected when the scientists dissected the otolith, and they were like, this otolith looks a bit translucent and it has weird shapes, and yeah. But it's still very novel work, and they don't really know what's going on with the formation when they swim in a particular direction or not. Yeah, great question.
And which species has the biggest otoliths based on what you have seen so far?
I'd like to clarify that this is not a real otolith, but it's an otolith from, it's a model otolith that I have. The biggest otolith I've seen are from snappers. So I think my biggest otolith is something like 3 centimeters in length. So it's also from a snapper, and it can be found in our local markets. So commonly used in our curry fish heads, actually. Yeah.
This question is also related to the shapes of the otoliths. Do the different shapes or any other properties of the otoliths indicate the hearing ability? Like, do some shapes mean that the fish have sharper hearing than others?
Based on my understanding, hearing varies between family, and I would imagine that the capability of a fish to hear varies between the different groups that they are in. But I can't remember off the top of my head which group has got better hearing than another group. But I would assume that I would think that there are differences in terms of hearing capability and the otolith. Yep.
I'm pulling out one of the earlier questions.
Yep.
Can you explain why one growth band corresponds to 1 year of age in fishes?
1 year of age. So it's, okay, let me pull up this slide. So 1 year of growth would represent 1 opaque and 1 translucent band. So this represents the fast and slow growing season. So when you're growing really quickly, the density of your deposition of calcium carbonate material is much quicker. So you would form a translucent band. And once you hit winter, for example, it becomes a more dense region because you are depositing material more slowly in these conditions. So it produces this very dark opaque zone, as you can see over here.
Okay, and then I think we can move on to questions related to conservation.
Yep.
This is from Josh. Can otoliths help in creating a fish quality index that factors in sustainability, taste, texture, and nutritional value?
This is, let me try to think. Based on my understanding, what we ultimately want to achieve is getting or extracting the information from the otoliths and then feeding this information into managers or people that manage the fish population. So making sure that we manage our fish populations well, whether we can get a proxy for good quality fish, I'm not too sure whether we can do that, but at the very least, we want to try and make it sustainable by getting a better understanding of how the stocks or how the fish populations are doing right now.
Okay, another question related to conservation. How has the progress with studying otoliths in Singapore have been so far? And can you share some of the findings of your research on fish population in Singapore?
Oh, okay. Yeah, sure. What I found, so I've been collecting fish from a broad region across Southeast Asia. And what I found was that with increasing temperature, the 2 snapper species that I'm looking at, they seem to be positive. There seems to be a positive relationship between their growth and temperature. So meaning that future climate change may not affect them detrimentally. However, what I found was that there are differences between the 2 species that I'm looking at and between the life stages. So while the adults might be doing okay under future climate change scenarios, juveniles may be affected detrimentally with increasing temperature. So good for adults, bad for juveniles, but it adds to our understanding of conservation and fish ecology.
Following up on that
Mhm.
question, are there any challenges you have faced while studying fish otoliths? Like, are the structures too small to extract information from it, etc.?
I think it's a major challenge in starting a field that's been quite established in Southeast Asia. So first, we've got a lot of different species that we could study. Some otoliths from certain families are much smaller than others, as you would see from, for example, your pufferfish, which has got otoliths which are 0.5 millimeters. That makes it really difficult to extract. And I think it's also very difficult to count the number of bands in it. So you need to use really specialized equipment to section the otolith to get a sense of the number of rings. It's easier for the larger species, I would think, which is what I'm working on, for example, snappers and groupers, commercially valuable. Another challenge in working in Southeast Asia is related to the supply chain in which we import our fish into Southeast Asia or into Singapore. So we import up to 90% of our fish into Singapore via our fishery port. And often when you go to a fish market, you ask the fish supplier, do you know where this fish is from? And they would just tell you Indonesia or from the ocean. So which is very difficult for us to actually implement sustainable practices because it's between nations and we need to go back to the source of where fishermen are actually catching the fish. So I think that's really challenging in trying to establish where or which region the fish are from, but we've got good working relationship with suppliers which know the fishermen personally and work with them for over decades. So that establishes a bond and they are happy to work with scientists to actually get a better understanding of the impacts of fishing and climate change on fish populations.
Okay. One interesting question. How
Yep.
were you first interested in otoliths?
That is a very good question. I know it's a very niche field and like how would people know about otoliths in the first place? So I studied overseas and I was in a lab that did a lot of fish research. And one part of the fish research was doing animal behavior in experiments. And the other half of the lab was doing otolith research. And there was a project that sort of integrated both the research on behavior and what they do with their lives using experiments and otoliths. So getting a better understanding of what's going on with the fish. And so I sort of got hooked, tried a few things, and found that these structures have very unique properties, which made it interesting for us to study.
Okay, it's about 8:45. I have just one last question that I think some of the participants might be curious to know as well. Have you seen any cool stuff while dissecting some of the fishes other than just the otoliths?
Yeah. I've seen a lot of cool things while dissecting otoliths. So I actually prepared a slide just in case this question came out. So what I found was I've dissected otoliths and I found a small fish being eaten by a larger fish. So just in the top left-hand panel, left-hand image, it's a very tiny fish that was swallowed and then it was caught right after. In the bottom left, I found a half-beak, which is commonly caught species, commonly found species in our local waters, and yet this huge parasitic crustaceans in its gills. So they're clinging there and they this fish is, the head of this fish is about maybe 7 centimeters or 7 or 6 centimeters. And these parasites were maybe 1 centimeter. So the whole gills were like, there was a huge crustacean on the gills which were parasitizing on them. I've also found a shark in the esophagus or the digestive tube of a snapper. So it's a baby shark, and I think it was a 5 kilogram snapper, so something like a really big snapper which was eating a shark. So you can see the weird and the mysterious. And during one of my visits to a wet market in Singapore, I pulled out a baby stingray from a pregnant female as well. So that's all the interesting things that you can see from doing dissections. You can see what they eat as well. For example, you'll see crabs or fish or half digested items in their stomach as well as their gills. Yeah.